Okay, so if you check course resources on the left-hand side, I've put some websites that I found and websites and apps that people have brought to my attention. There's some helpful things here. Virtual labs. This is pretty cool. If you click on the link, it has. It doesn't have just microbiology. It has immunology, chemistry. There's some more advanced labs. Just move on down to what you would like to learn about. Here's immunology. You can learn about different methods. Here's the micro lab. Selective and differential media for identifying organisms. So you want to learn more about that. And it gives you some background and the purpose and the principle and some examples, and it talks about it. Mannitol salt auger. It's got stuff up here if you want to take a quiz. You can take a quiz on the information. Animation. This has like it's free sign up, free login. Not all of them. Let's see. Require it. I was able to get into the animation and the simulator upstairs without logging in, so maybe it's different depending on what you click on, but it's like a virtual performance of the test in a lab, so you can see what it looks like. What else? Micro website. This has quizzes and exams on a bunch of different information. Here's the American Society of Microbiology. It's got a gallery that you can use if you don't remember what Staph aureus looks like, or say you don't remember what the CAMP test is or what it looks like. Here's a CAMP test. It's got strep agalactica and strep pyogenes on a plate. This is Staph aureus. Here, this is group B strep, or strep agalactica, and it's CAMP positive, so you have the arrowhead. This is group A strep, or strep pyogenes, and it's CAMP negative, so you can see what that looks like. So all different types of tests: citrate, coagulase, gram stain. There's a website if you want to make flashcards, not by hand, but through this app or website that they have. You can make them here and then print them out. If you want to make your own quizzes, there's a quiz website. Some websites, just more quizzes. So check that out, definitely. Pictures. Here's app lists. Most of these apps are free. Some of them might cost money, but there's all different kinds. There might be chemistry, blood bank, micro, just general micro or general lab apps. So check them out. Let's see here. Okay, so chapter 17 is your gram-negative cocci. And it's a pretty easy chapter, probably one of the easiest chapters that we'll talk about. There's only 3 organisms that we're gonna learn. Majority of your Neisseria organisms are normal flora. There are a couple of pathogens. Does anybody know some pathogens? Neisseria gonorrhea, Neisseria meningitidis. So those are your primary pathogens that we're going to talk about today. And then we've also got Moraxella catarrhalis. So make sure you take a look at these objectives as you're studying, getting ready for the test. Here's a case in point. So we've got an 18-year-old sexually active college student. She went to the student health complaining of pain and redness and swelling on both wrist joints and her left elbow. She's got a casual history of unprotected sex and 3 or 4 men during the last 4 months. She's denied any kind of vaginal discharge or abdominal pain, but she does recall having a rash on both of her arms. So they took an aspirate from the left wrist and sent it for a gram stain and a culture. On the gram stain, they saw many PMNs, or lots of white cells, right? Indicates bacterial infection if we see SEGs. And then rare intracellular and extracellular gram-negative diplococci. At 24 hours, many small tan colonies were visible on the chocolate agar, but there is no growth on the sheep blood agar or the anaerobic plate or the McConkey plate. So what do those results tell us here? We don't have any growth on McConkey. Does that correlate with our gram stain? What does McConkey grow? Gram-negative bacilli. And does that correlate with our gram stain? Do we have any gram-negative bacilli in our gram stain? No, so that looks good. What else? We've got no growth on our anaerobic blood plate. What does that mean? It's not an anaerobe, right? It's only growing on our chocolate agar. What does that tell us? It's probably a fastidious organism because it's only growing on chocolate, not growing on the sheep blood agar. Something else is this intracellular and extracellular gram-negative diplococci. That's a notorious gram stain for Neisseria gonorrhea. So based on those characteristics, that's the identity of the patient's infection. What are some risk factors for acquiring this organism? Unprotected sex, right? Let's see. And then we're going to talk about some sequelae, which are like secondary diseases almost, or things that, diseases or manifestations of a clinical disease that happened after initial infection. It's because the patient wasn't treated and the disease continued. What's the importance of identifying the organism? We're going to talk about some complications. And then what's the significance of beta-lactamase testing? What's beta-lactamase testing? Anyone remember? We talked about it with antibiotic sensitivity testing. It's an enzyme that the organism might produce that breaks down beta-lactam antibiotics. So it would make beta-lactam antibiotics ineffective. So what's a beta-lactam antibiotic? An example? Anybody know? Penicillin, right? It's a primary treatment for gonorrhea is penicillin. So if we test this gonorrhea organism with our beta-lactamase test and it's positive, what's going to happen if we treat the woman with penicillin? She's going to get sicker because it's not going to be effective. All right. Okay, so some general characteristics of Neisseria organisms. They're aerobic. I tried to underline what's important. I don't memorize whether an organism is aerobic or anaerobic. I know when it's anaerobic, but I don't learn, is it facultative anaerobic, is it aerobic? You're probably not going to have a test question that asks, is Neisseria aerobic or anaerobic or facultative? Let's learn some more important things here. Gram-negative diplococci. They're like kidney beans, usually, for gonorrhea. Anyways, 2 kidney beans facing each other. They're always in pairs, Neisseria are. They're oxidase and catalase positive. So these are really rapid tests. We've done catalase a couple of times. And oxidase is basically touching a colony with a swab and putting the oxidase reagent on there, looking for a blue color or a purple color. Or maybe using a filter paper that has the oxidase reagent and smearing some colony on there. Does it turn blue? So these are really rapid tests that we can do to really quickly identify or categorize our organism. Okay? So if it's not catalase or oxidase positive, we know it's not a Neisseria. Their usual flora in the upper respiratory tracts and the genital tracts. There are some Neisseria meningitidis can actually be normal flora in some people. It doesn't necessarily cause symptoms or disease, and these people are called carriers. Your primary pathogens in this section are your Neisseria and your gonorrhea and Neisseria meningitidis. So we're going to learn how to differentiate those pathogens from your usual flora. That's going to be there routinely. So this is just showing, this is the family Neisseriaceae. They're all gram-negative cocci, diplococci. They might be coccobacilli or rods. We're not going to talk about some of these. We'll talk about Eikenella in a little bit. We'll talk a little bit about Kingella. But for the most part, we're going to focus on Neisseria, gonorrhea, and meningitidis and how to differentiate those. Some virulence factors for Neisseria. These are factors that they have that allow them to cause diseases in humans. And there are more other than what's listed on this slide, but this is what I want you to know. They have capsules, so that allows them to evade phagocytosis or resist phagocytosis. And we'll also see a clearing around the organism on our gram stain. They have pili, which are hair-like structures on the surface that do a few things. They can attach to host tissues. The urogenital tract and the respiratory tract, they have those ciliated epithelial cells, right? They've got a lot of mucus, a lot of movement going on. So they need to have something to latch onto those mucous membranes so that they can set up infection and replicate and cause disease. And then these also help to prevent phagocytosis, and they can exchange genetic material from one cell to another cell. So Neisseria gonorrhea is the first organism we're going to talk about. Humans are the only natural host for this organism, and it's not normal flora for Neisseria gonorrhea. Neisseria meningitidis can be found as normal flora, but not this guy. Some history, gonorrhea is means flow of seed. I think it's French. You might also hear it called the clap, and this is from the clap. I'm probably not saying it right. Clapior. Does anybody know French? Nobody? I took Spanish. Anyways, that's where the organism comes from, and that. Let's see, it's transmitted through sexual contact. It is a reportable disease, so if we do isolate it in the microbiology lab, we do have to report it to the health department, and they keep track. The reservoir, there are some asymptomatic carriers, so there's people that carry it that don't manifest infections, but it's not normal flora in those people, so it's kind of contradictory. We think of usual flora as organisms that are there naturally and that don't cause disease. Gonorrhea can be present in especially females and not cause disease or symptoms, but it's not supposed to be there. The clinical infection that we see, we typically see it in the urogenital tract, but we can also see it in the pharynx and the conjunctiva of the eye. So this is primarily caused in newborns during birth through the vaginal canal from a mother who is infected with gonorrhea, and she passively transmits it to the newborn. There are some other kinds of infections that gonorrhea can cause. We can see disseminated infections. What is that? What's disseminated mean? Disseminate something, like disseminated papers. They spread them around, right? Usually happens after a localized infection, say maybe a urinary tract infection that gets into the bloodstream. Then it's able to disseminate to other organs, right? So there are some disseminated infections where gonorrhea kind of gets out of its localized infectious area and spreads to other parts of the body. Gonococcal urethritis is a common cause of disseminated infection. So it might start in the genital tract, but then for some reason it prefers to localize to joints and cause arthritis, and then the anal canal for obvious reasons. Disease in the male, transmitted through sexual contact, just like in females. 95% of men do show symptoms if they're infected, whereas in women, that number is a lot lower. Symptoms, we've got painful urination and a discharge, and some other complications, epididymis and urethral stricture and prostatitis. So in females, 50% are asymptomatic. And typically see it in the endocervix. Symptoms, if they are showing symptoms, got painful urination, cervical discharge, and abdominal pain in the lower abdomen. Some complications here, if we don't treat the disease or if it goes on without treatment or successful treatment, we can see pelvic inflammatory disease, sterility, ectopic pregnancies where the fetus is developing outside of the uterus, and then hepatitis. Okay, so infections in other sites, there's extragenital infections, and then diseases in newborns. So when the newborn is born of a mother through vaginal birth who has gonorrhea, a lot of times they'll get the gonorrhea in their eyes, and we can stop infection just through some eye drops. Some disseminated gonococcal disease, bloodborne dissemination if it gets in the bloodstream. We'll see fever, bacteremia, and rash on the extremities. Kind of sounds like our case, right? Our case study. One thing to note is we usually use blood cultures to grow bacteria, right? If it's in the blood, this is how we diagnose it. But it has this ingredient, the SPS ingredient, and it inhibits the growth of Neisseria gonorrhea. So you can't use usual blood culture media to grow it. And then we can have gonococcal urethritis. Arthritis, not urethritis. Some clinical samples. So if someone's presenting and the physician's suspecting gonococcal, there's a variety of different clinical samples that you're going to collect, and it really depends on the presentation of the patient. Urethra in the males, the endocervix in the females, if they're presenting with those kind of infections. Rectal culture, oral or pharyngeal culture, eye culture, joint, blood fluid. You should, if it's from one of these other sites, they should notify the laboratory, because it's not common. We're going to talk about some selective media that we use to grow gonorrhea. It's not common to use this selective media from a throat culture or an eye culture. So it's important that we're notified if they do suspect this organism so we can add it to the routine media. Now, genital sites, we're always going to set up something that will isolate gonorrhea. So these are the different types of media that we might inoculate. And these are transport media. So transgrow or Jim-Beck is a big one. And basically the nurse or physician inoculates the transport media right after collection. Remember, it's a fastidious organism. They're sensitive. So we've got to inoculate the transport media right after we collect it. And if you notice, that little round pellet is something that will convert oxygen to CO2. So once you inoculate your media, you stick it in the bag with the pellet in there, and it'll convert it to a CO2 environment, because gonorrhea is a capnophile that likes CO2, and then transport it to the lab, and they'll do their thing with it. If you can't put it on some of these transport medias, then you want to use like an Ames media with charcoal, and transport it to the lab as soon as possible. We want to plate it within 6 hours. So here's some microscopic morphology of gonorrhea. Gram-negative diplococci. It's real hard to see here the kidney bean shape. Intracellular, so it's inside white blood cells. Here's a good one here. I don't ever say intracellular. I rarely say gram-negative cocci unless I'm absolutely sure. Because when physicians hear intracellular gram-negative diplococci, they think of gonorrhea. One important note here is the presence of white cells with no bacteria indicates non-gonococcal urethritis. So inflammation of the urinary tract, but it's not caused by gonorrhea. So that might be a test question. If you see white cells but no organisms. All right, so that's a microscopic morphology. How do we grow it and what's it look like on the plate? Here, real important, it grows on chocolate agar but not on sheep blood agar. Not usually. We can't say 100%, but for testing purposes, gonorrhea doesn't grow on sheep blood agar. They can do whatever they want in the real micro lab, really. It's incubated at 35 in 3 to 5% CO2 because it is a capnophile. One way you can do that if you don't have a CO2 incubator, I don't have one in the lab, so I use a candle jar. So you might have seen me, or you might have set one up where you just put your inoculated media in a jar. It doesn't have to be a high-dollar jar. It can be a big pickle jar. And put your candle in there and light it and seal it up. And when the candle goes out, that means all the oxygen was converted to CO2, and you've got your good atmosphere. Some selective media that you want to use that inhibits the usual flora that's present in the genital tract. Makes your life a little bit easier to spot these pathogens. We've got your Thayer-Martin, your modified Thayer-Martin, New York City agar, GC-Lect agar. So if you see growth on your chocolate agar and no growth on your sheep blood, and you have growth on your modified Thayer-Martin, it suggests that you have one of these pathogens, Neisseria gonorrhea or Neisseria meningitis. You can't confirm that. You have to do biochemical testing to make sure, but it's a strong suggestion. So what it looks like on colony morphology on modified Thayer-Martin and chocolate is just this gray, small, translucent, might be semi-opaque, slightly raised. Okay, so once you've got a colony, you think it's gonorrhea, what are you going to do first? You want to do your rapid testing like your catarrhalis and oxidase, right? And what's the reaction for Neisseria for those two tests? Positive, right? All right, so we've got something that's positive for oxidase and positive for catalase. Here's an example of both. This is an oxidase disc, and you probably just put a drop of the reagent on there and smear some colonies on there. Or you can put the oxidase reagent directly on the growth. I don't like doing this because then you contaminate your plate. So I'd rather use a disc, or, a swab, or you can use a coffee filter and put some oxidase on a coffee filter and smear some on there. Some other confirmatory testing that we can do for definitive identification to confirm the identity. A superoxyl, which is a 30% hydrogen peroxide solution. It's performed just the same as the catalase, except it's a higher concentration. It's a toxic reagent, so I don't have it in the lab. Carbohydrate and gonorrhea is positive. All other Neisseria are negative. So that's a quick, easy test. Carbohydrate utilization. What sugars does it ferment? Chromogenic substrates. We'll do the Neisseria enzyme testing kit in lab today, and that's a chromogenic substrate. We're going to look for certain enzymes that gonorrhea will produce that other Neisseria don't produce. Immunologic assays, like agglutination methods, using an antibody to identify a colony. If you use an anti-gonorrhea antibody and you mix it with your colony and you see agglutination, that tells you you have gonorrhea, right? And then there's nucleic acid testing or molecular techniques that we'll talk a little bit about just so that you're aware that they exist. So cysteine trypticase agar, CTA, these are just carbohydrate tubes, 1% concentration of glucose, and then you'll have another tube with maltose, and you have another tube with lactose, maybe another one with sucrose. So you inoculate these tubes with your organism. It's got a phenol red pH indicator just like your TSI, and you incubate it in ambient air. And if the sugar is fermented, we're going to see a decrease in the pH, and your color change will turn to yellow. Same principle as other carbohydrate tests. Your chromogenic substrates, you're detecting an enzyme that's able to break down a colorless substrate, which produces a color. You want to use isolates from selective media only, so you don't want to use chocolate agar isolates. And that just improves the accuracy of the test. And then there's multi-test methods that will combine biochemical and enzyme tests. There's agglutination methods, fluorescent antibody testing, so you can use a fluorescent tag on an antibody and test it against maybe some tissue or a smear. And if the organism is present, it'll fluoresce a certain color. There's MALDI-TOF, which is some automation that's coming up and coming. Nucleic acid testing is very common. A lot of physicians will prefer the nucleic acid method over these culture methods. It's very rapid. It's very sensitive. There are some disadvantages. Let's see. Can't do sensitivity testing. Let's see. It's not approved for certain sites. I don't think you can use it on children. So diagnosis of children, this is a good test question. If you do isolate gonorrhea on a child that's over neonatal, so it's past that time frame from where they would have caught it during birth, it suggests sexual abuse. So in that case, these are the scenarios that have to occur in order for you to call it gonorrhea. Number one, you have to exhibit the typical colony morphology on selective gonococcal media, like your modified Thayer-Martin in your New York City. It has to have that notorious gram stain, gram-negative diplococci. It's got to be catalase and oxidase positive. And then you've got to confirm it using two different methodologies. So your chromogenic substrate assay and carbohydrate fermentation. That would be two different methods based on two different principles that you can use to confirm that it's gonorrhea. And then you cannot use non-culture methods like molecular methods in children. And then finally, antimicrobial resistance. We're not going to talk about a lot of things here. This is the only thing we're going to talk about, but is the nitrocephin disc or cephinase disc for beta-lactamase production. So we smear a little bit of the colony on this disc, and if it turns pink, it means that the organism produces beta-lactamase. So here's an example of what's going on. Here's penicillin, and it's got this beta-lactam ring, and all beta-lactam antibiotics have this common ring structure. And if the organism produces this enzyme that breaks down beta-lactamase, it'll break that ring, and that changes that drug into another substance that isn't an antibiotic. It makes it ineffective. So make sure you know this test for the exams. We'll test it. Well, Neisseria gonorrhea gets this test, and several other organisms like Moraxella catarrhalis will commonly get this test. All right, so Neisseria meningitidis. Like we said, it can be normal flora in some carriers. It's transmitted through respiratory secretions, so coughing. It primarily affects infants, adolescents, and immunocompromised. And here's a particular population that's at risk for this organism. Make sure you know this for exams.
Crowded conditions, people that live in crowded conditions, like dorms in colleges. That's why you're supposed to get this vaccine when you go to college, right? We're at susceptible risk. Military recruits, they're living in big barracks, very crowded. So these people are definitely at risk for this organism. And it's very fatal, 25% even if you do get treated. It usually can cause mild symptoms, but there are cases where it becomes very serious. It gets into the bloodstream. It can get into the central nervous system. Let's see, some red flags that someone has meningitis, serious headache. Somebody heard of this before? If you go to the ER, really bad headache, they'll do a spinal tap, look for meningitis. I think of this organism when ER. Anybody watch that show way back in when with George Clooney? Well, there was an episode where college students there was an outbreak of meningitis, and everybody was coming in with really bad headaches, and they're doing spinal taps. And then when I was in my college days, I had several friends that would go to the ER with bad headaches, and they would do spinal taps, and it's because they're looking for this organism. Stiff neck, that's a big clue. If you have a test question that says stiff neck and headache, you want to think of meningitis. Nausea and vomiting, fever. Meningococcemia is, you want to think of rashes. This baby has petechiae all over its body. It's like red, pink, blotches on the skin, on the mucous membranes too. Fulminant disease is when it spreads through the entire, not spreads through the entire body, but it's very acute, very aggressive type of the disease. And these are the adrenal glands. I don't know what they're supposed to look like. I guess they're supposed to be pale pink. But they start hemorrhaging. And there's some other complications. So the specimens that we collect for Neisseria meningitidis, number one is your spinal fluid. You can also get blood, and of course it's a respiratory disease, so you want to get nasopharyngeal or maybe aspirate. If there is more than 1 ml, then you need to centrifuge the spinal fluid, and you want to use the bottom ml to inoculate your plates and make your gram sing. That concentrates the organism in the very bottom of your tube, right? Again, you don't want to use your normal blood culture systems because it has that ingredient in it that inhibits the growth of these pathogenic Neisseria. If you're going to culture it, it does grow on sheep blood and on chocolate agar. And then you can use your selective agar like your modified Thayer Martin. It'll grow on that also. Incubate at 35 in a capnophil environment. And then this is a BSL-2. You want to work under the hood with it because it's highly transmissible and fatal. So here it is, gram-negative cocci, and here it is growing on both the sheep blood and the chocolate agar. I included this because you haven't had body fluid yet. You'll have it in the summer. So this is helpful. It kind of differentiates meningitis when it's caused by bacterial source or a viral source or a fungal or a parasite. So I just wanted to highlight a few things. The cell count is usually very elevated when you have a bacterial source. So say they get a spinal tap of some spinal fluid and you do a cell count how many white cells, how many red cells are present in that sample. So bacterial source, it's going to be elevated. And then you'll do a diff and you find out what kind of cells are there. Okay, so primarily neutrophils are going to be predominant. All right, so when you hear neutrophils or SEGs, you think of bacterial infections, right? What do you think of when you hear lymphocytes? What kind of infection? Viral, right? What about eosinophils? You want to think of parasites, right? So,
these are important too, protein and glucose. So if it's a bacterial infection, your glucose is going to be decreased and your protein is going to be increased. And that's because the bacteria are eating up the glucose. They're using it for energy. And anytime something is living, it's producing proteins in order to live, right? So you have more things there that are living and functioning, so your protein is going to be more elevated than it should be. So if you see that, if you see protein high, glucose low, think of a bacterial infection. And that'll help you differentiate from other things. Might help you rule out a couple questions or answers on a test question. And then I just wanted to point out that there is a vaccine available for Neisseria meningitis, and it's given to populations that are at significant risk, like your military, your college students, patients that have had their spleen removed. Moraxella catarrhalis. This is a normal commensal flora, normal flora in the respiratory tract, but it can cause infections if it's given an opportunity. Typically what we see is the sinusitis and otitis media, so basically a sinus infection. Okay, we can see some pneumonia, endocarditis, and meningitis, but I want you to note the sinusitis and otitis media in children. We see it a lot in kids. And there are some predisposing factors. Anytime you have an opportunistic pathogen, very old, very young, immunocompromised, people with low seg count, neutrophil count. All right, your specimen collection for otitis media, you're gonna go to the source of the infection. So your nasopharyngeal, your sinus aspirates. If it's an ear infection, you'll get an ear effusion or swab or aspirate. Sputum, if it's a pneumonia. Bronchial aspirates. On your microscopic morphology or gram stain, see how big these are compared to gonorrhea? Gonorrhea was very small. Moraxella is very fat, very large gram-negative cocci. And its colony morphology, it does grow on sheep blood agar and chocolate agar. So it's not fastidious. We can smooth, opaque, gray, white colonies. So it's not very unique in that sense, but it does have a hockey puck motility. So if you go to touch the colony might slide. The whole colony stays intact and might slide across the agar. So you want to think of him if you hear that description. Older colonies kind of have a wagon wheel appearance. So maybe at 48 hours of growth, we see this wagon wheel appearance. You might see it better in your book, but it's kind of raised in the center, and you can almost see like striations around the raised center, and it looks like a wagon wheel. So think of this guy if you hear wagon wheel. How do we identify it? Well, we're going to do our rapid test. Since it's a gram-negative cocci, we want to do a catalase and an oxidase, and it's positive, just like your Neisseria. One way to differentiate it, it's asaccharidic, so it does not ferment any sugars. Okay? DNA is positive. Very few organisms are DNA is positive. So this, if you see DNA is positive and gram-negative cocci, you should know right off the bat it's Moraxella catarrhalis. Another helpful butyrate esterase, or the MCAT disc. We'll do this in lab this week. It's the only organism that produces this butyrate esterase enzyme. And if you smear some of the colony on the disc and wait a couple of minutes, 2 minutes, and it turns blue, it's Moraxella catarrhalis. So easy test questions here. Okay, this is from one of your charts in your book. I kind of broke it down into what I want you to know. So definitely know the glucose fermentation reactions, and this is the enzymes that we test for on the chromogenic substrates that we'll do in the lab. So we'll test for 3 enzymes.
And then we have our DNase and butyrate esterase and a nitrate down there. So we know that Neisseria and Moraxella are both catalase and oxidase positive, so it's positive all across the board here. Growth on modified Thayer-Martin or New York City, that's media, selective media for gonorrhea and meningitis, right? So your Neisseria will grow on that. Neisseria lactamica is usual flora. And it can grow on there. Usual flora and saprophytic Neisseria, which is just normal flora Neisseria, do occasionally grow on our selective media. That's why we have to do our confirmatory tests. Moraxella will not grow on it. All right? Kingella, and I just included that just so you can kind of see the difference. It's gram-negative bacilli, so your gram stain will help rule it out. If you do a nitrate, that might help rule it out. Let's see, superoxyl. That's your 30% hydrogen peroxide. It's like your super catalase test. Only gonorrhea is positive. So that's a quick and easy way to recognize it. Then we have our glucose, maltose, and lactose fermentation. What do the organisms ferment? So if you notice, Neisseria lactamica, it's our usual flora, non-pathogenic Neisseria. It ferments all the sugars, right? It uses anything it can get its hands on, it will ferment. All right. Also, it starts with an L, and lactamica starts with an L. So that's an easy way for you to remember. If it ferments lactose, it's usual flora, or lactamica. Now, here's maltose, starts with an M. Notice Neisseria meningitidis ferments glucose and maltose, but not lactose. So if I see it ferments maltose, I know that it's meningitidis. And then gonorrhea will only ferment glucose. G, gonorrhea. Okay? That's how I remember it. So you can easily differentiate between the Neisserias. Notice Moraxella catarrhalis, we said it's asaccharolytic. It doesn't ferment any sugars, doesn't use it. Now these are the chromogenic substrates. So we're testing for production of a certain enzyme. Okay? First, we have gonorrhea. Gonorrhea will only produce this hydroxyprolamyl amyl peptidase. And don't worry about memorizing this whole word. I kind of picked out the PRO. I look for that PRO. I think prostitutes and gonorrhea, right? So that helps me remember. And then we have glutamyl amyl peptidase. I couldn't really figure anything out with that. Galactosidase. This has lactose in it, doesn't it? And it's only positive for lactamica. So that helps me remember. So if I can recognize that pattern, I can remember that meningitis is positive only for that oddball enzyme that I can't remember. Okay? And then we said DNase and butyrate esterase are used for what? Moraxella catarrhalis. He's the only guy that's positive for those two. So that helps. Okay? All right, let's take a quick break, and then we'll talk about fastidious. That wasn't too bad, was it? Not too bad. Okay. So this is the first part of our gram-negative bacilli that we're going to talk about. So these are your fastidious organisms that only grow on chocolate agar, right? And for the most part, they're tiny coccobacilli or gram-negative bacilli, very tiny. Okay, take a look at your objectives. I'm not going to talk about all the organisms in this chapter, and I'm not going to talk about a lot of them as comprehensively as we're going to focus on Haemophilus and gonorrhea. There's some things that I just kind of want to point out to you for you to recognize. So let's look at this case in point. 59-year-old female stroke victim. She's confined to a nursing home. Let's see. Her admission, she complained of slight respiratory ailment. She awoke 3 days later with a stiff neck and severe headache. Ooh, what does that make me think of? Meningitis, right? Can other things cause meningitis besides Neisseria meningitis? Oh, yeah. Okay, so because of her high fever and the other symptoms of meningitis, she was transported to emergency room where they did a spinal tap. So spinal fluid was cloudy. Oh, that's a good hint that it's infectious. Not supposed to be cloudy, it's supposed to be like water, clear, see-through. Elevated protein, decreased glucose. Oh, what does that tell me? Bacterial infection. If that was me working in chemistry, I'd start to get worried, and I do. Because and we've had one instance where we had a patient come in with an elevated protein and decreased glucose, and they thought she had Neisseria meningitis. So everybody in the lab that was working on this fluid had to take antibiotics. Really high whites, 420 white blood cells per mil. That's very, very high. 92% are SEGs, PMNs. That's high too, right? It makes us think of bacteria instead of a viral infection. All right, so they did a gram stain on the spinal fluid, and we saw many white cells and many small gram-negative bacilli. Can I not be worried now? Yeah, because it's not Neisseria meningitis. It's something else that's not as serious as meningitis. So that would calm my nerves. Some appeared like a coccobacillary with clear halos. What does that mean? Encapsulated. A lot of your organisms that have capsules are able to cross the blood-brain barrier very easily. So someone might start out as being septic or bacteremic, have bacteria in their blood, and it can cross the blood-brain barrier and get into their spinal fluid easily because of that capsule. Let's see. Cultures of the spinal fluid and blood produced a heavy bacterial growth on chocolate agar, but not sheep blood or McConkey. Well, that clues me into two things here. We've got growth on chocolate, not on sheep blood. What's that tell me? It's a fastidious organism. Also, it's a gram-negative bacilli, right? Is it supposed to grow on McConkey? Supposed to, but it's not growing on McConkey. So that puts it in this category, a gram-negative bacilli that's fastidious. Okay? So if it's not growing on McConkey, it belongs in this group. Everything else should grow on McConkey. Okay, so predisposition to meningitis. She's not up and moving around. She's in a nursing home, which is crowded, right? Sick people. Respiratory infection that might produce a capsule, can get in her bloodstream and cross the blood-brain barrier, or even skip the blood and just travel to the spinal fluid through the respiratory tract. Is a vaccine available for this organism? Primary causes of meningitis in this age group. We'll talk about meningitis in very young, 3-month-old to 6 years, and we'll talk about meningitis in older populations, like your 60s and your 70s. So different organisms will cause meningitis in your older population than you would see in your very young population. So the age of the patient can clue you into a certain kind of organism. And then some characteristic of the microscopic morphology and the growth patterns that help you lead to this presumptive identification. So just from this, I'm thinking Haemophilus influenza. So some general characteristics about these organisms. They're fastidious. They need special nutrients in their media in order to grow. They're small gram-negative bacilli. We'll talk about other gram-negative bacilli. They're big, like boxcars, boxy, real easy to see. Unable to grow on McConkey agar. They're gram-negative bacilli. They should grow on McConkey agar, but they're fastidious. There's something that they're lacking, that McConkey agar is lacking. that it requires in order to grow. So obligate parasites of the respiratory tract. Haemophilus ducreyi is your exception to that. So most of these are respiratory tract pathogens. So Haemophilus species is the first one we're going to talk about. They're gram-negative, pleomorphic, which means you might see them as a coccobacilli. You might see them as rods. You might do a gram stain on that colony, and you might see two different morphologies. You might see rods, you might see coccobacilli. So that's where we get pleomorphic. They're tiny coccobacilli in direct smears. You notice when we did gram stains, we had all that proteinaceous debris that was pink, and it was kind of hard to see your gram-negative organisms through that background material at first. These guys are super hard to see. It requires some experience, and even some seasoned techs can miss it on smears. Your biochemical reactions, oxidase, catalase, and nitrate positive. So same as your Neisseria species, except for that nitrate. They're not, Neisseria is not nitrate positive. But they're gram-negative coccobacilli. Most are non-pathogenic, but they can produce your opportunistic infections if they get the opportunity. So some of the organisms that we're going to talk about, the pathogenic, is the Haemophilus influenza, your Haemophilus aegyptis, and Haemophilus ducreyi. So first thing we need to talk about before we talk about Haemophilus influenza is growth factors. And this helps us determine which Haemophilus it is, based on which growth factor it needs in order to grow. So Haemophilus, the word means blood-loving. It loves blood, and X and V factors are components of blood. So X is the hemin, and your V factor is this nicotinamide adenine dinucleotide, or NAD. So it needs this X and V factor. Now, sheep blood agar only contains the X factor. Chocolate agar contains both the X and the V. So that's kind of why we see these fastidious organisms only growing on chocolate, because it has both factors, whereas sheep blood agar only has the one. Okay? All right, so if a Haemophilus species requires both X and V factor, what media will it grow on? Chocolate, because it has both, right? What if it only requires X factor? Sheep blood, will it grow on chocolate? Yes. What if it only requires V factor? Only grow on chocolate, right? All right, so these are your Haemophilus species that you should be able to differentiate. We're not going to talk about these individual ones and or the diseases that they cause, but I want you to be able to differentiate between them. Okay, so these Haemophilus influenza and Haemophilus hemolyticus both require X and V factors. So Haemophilus influenza is your main pathogen that we'll talk about. Haemophilus parainfluenza and Haemophilus parahemolyticus only require the V factor. If I can remember that para is V factor only, then I can spot these organisms and figure out which growth factor they need pretty easily, right? If it only requires X factor, Haemophilus ducreyi, he's our exception, okay? And then finally, how do you tell the difference between Haemophilus influenza and Haemophilus hemolyticus? Well, hemolyticus is hemolytic on sheep blood agar, or horse blood, or rabbit agar. So if it has hemolyticus in the name, then it hemolyzes the blood agar. So if you can break the name apart, you can figure out which factors it requires. Okay? Pretty easy, right? Satellitism is a phenomenon that we see on sheep blood agar. Haemophilus influenza, does it grow on sheep blood? What factors does it need? X and V. And which factors does sheep blood have? just X. So will Haemophilus influenza grow on it? It should not, right? Well, here we have Staph aureus, and we've got teeny, teeny, tiny colonies kind of growing around the Staph aureus. See that? They're almost too hard to see. That's the Haemophilus. So the Staph aureus is beta hemolytic, right? It's lysing the red cells and freeing up the V factor that's available only inside the sheep blood, the intact red cells. So now that X and V factor is available, so the Haemophilus will only grow around the Staph aureus. Here's another example, and it's kind of hard to see, but your Staph aureus are these larger colonies. It almost looks like water droplets because they're kind of growing together, but your Haemophilus are these teeny tiny ones that are growing in between. All right, so these are the some characteristics about Haemophilus influenza. The virulence factor, again, it has a capsule. So if we recognize that it has a capsule, then we can kind of assume that it is a good cause for meningitis. It allows us, the capsule can allow us to serotype it, so we can use immunologic methods like using a specific antibody, a Haemophilus influenza antibody, and mix it with our suspicious colony, and if it agglutinates, we know we have Haemophilus influenza. And then it has, of course, anti-phagocytic and anti-complement activity. It can evade phagocytosis. So that allows it to set up infection. Clinical manifestations, so what do we see? Haemophilus influenza serotype B. We're not going to talk about serotypes. Just be aware that there are other strains or types of Haemophilus influenza out there. This one, it might be familiar because there's a vaccine against this particular serotype. And children are supposed to get this vaccine because it causes meningitis in that age group. And then we have some non-typable strains that don't have a capsule. And these guys are less, the infections are less severe. Usually just respiratory tract, and the infections stay localized to a certain area. Usually respiratory tract, bronchitis, sinusitis, pneumonia in the elderly, maybe some genital tract infections. That's the Haemophilus ducreyi is STD. Haemophilus aegyptus, we're not going to talk about this one. I just want you to know that it's the cause of pink eye. It causes pink eye or conjunctivitis. Haemophilus ducreyi is a sexually transmitted infection, and it causes a chancroid. I don't know if I'm saying that right. It's how I say it. In the genital region, and it has a very irregular edge, and it's very painful. Syphilis also produces chancroids, but they're not painful lesions. And then you can see these really enlarged lymph nodes in the genital, these lymph nodes here become enlarged and start draining. So if you want, you can take a look at the picture. I had included it in previous PowerPoints, but it's just unnerving in the PowerPoint. Yes, you're welcome. Specimen processing for this organism, for Haemophilus. Remember, it dies very rapidly. It's fastidious, right? Very sensitive. So we need to plate it within 10 minutes for maximum recovery. It's got to get to the lab quick. Now, Haemophilus ducreyi, you do want to clean the site, and you want to swab the base of the ulcer, which is in the center of the ulcer. Usually, when you swab lesions, you get the middle. You don't want to get you get the spreading edge, the outer edge. That's where like the infection is fresh. The inside is where there's a lot of dead white cells and dead organisms. It's not a good place. But for Haemophilus, you want to get the center. And then you can also get an aspirate. That's your ideal source. But you can aspirate the pus from the lymph nodes or the buboes. Anybody hear that word before, buboes? The plague. You'll see buboes in the plague because the lymph nodes will as well. Bubonic plague, exactly. All right, so here's Haemophilus. Only grows on chocolate agar, right? Haemophilus influenza. You might see it satellite on sheep blood agar. If you see these terms, man, you want to think of Haemophilus. Translucent, moist, convex colonies, mousy or bleach-like odor. I smell the mousy odor, and our strain in here, I believe, is pretty mousy. Encapsulated strains or strains that have capsules will appear mucoid. Oh, here's some more pictures. Haemophilus influenza, teeny tiny coccobacilli. I can't hardly see them here, can you? You can probably see them better in your book. They're better here. This is Haemophilus influenza too, probably from a culture plate because there's no white cells and there's no proteinaceous debris. So it's more concentrated, but very teeny tiny. And notice that we see them kind of inside white cells. They might just be laying on top of white cells. They may be intracellular. I don't know. Don't say intracellular gram-negative coccobacilli. Why is that? The doctor's going to think of gonorrhea, right? So I just leave intracellular out of the question. I don't know if it's laying on top of the white cell or if it's inside. I can't tell, so I leave it out, just to be safe. Haemophilus ducreyi is coccobacilli also, and it resembles a school of fish. I couldn't find a picture of it, but it's like they stay together. They're kind of all clustered together in a gram stain. So if you see school of fish or maybe railroad tracks or fingerprints where they're all kind of swarming together, you want to think of Haemophilus ducreyi. And they'll use that terminology on your test questions. I will, and the Board of Registers will. Okay, so if we suspect it's Haemophilus, how do we identify it? You want to take the colonies from the initial isolation of Haemophilus. You don't want to pick up the growth media because, of course, that could influence your testing. Let's see. So here, for this, we're going to interpret the growth pattern. If we figure out whether it needs X factor or V factor or both, okay? So we're going to take a little bit of the colony and we're going to put it in a broth and we're going to mix it up. We're going to make a suspension and then we're going to plate it on a nutrient agar or a Mueller-Hinton agar plate that's designed for Haemophilus. And we'll do a little bit of this today. I don't have X or V strips, but we'll do antibacterial testing just so you can kind of see what it's like. And then you put the strips on the media and you incubate it in CO2 because it's a capnophile, 18 to 24 hours, and then you interpret the growth pattern. So you want to see where the organism is able to grow. So these strips are impregnated with the factors. So here on this example, we've got growth only between the strips, right? So which factor does it need? It's kind of leaning towards the X, but it's between the two. So when you put these strips on, the X and the V factor spread out in the agar. So the organism will only grow where the factors are provided. So Haemophilus influenza and Haemophilus hemolyticus both need X and V. So they will grow only between the two factors, or between the two discs. This one, we've got growth, see it's on both sides, whereas on the previous one, it's not on this side where there's only V, and it's not on this side where there's only X. It's only between. Oops. This one, it's surrounding the V factor, where the V factor was disseminated through the agar. So organisms that just need V factor are para-influenza and para-hemolyticus. They've got that para inside their name, so we know they just need V factor. Does this make sense to everybody? Okay. Okay, well, what if it just needs X factor? Haemophilus ducreyi is our exception, right? So it will only grow around the X factor. We got growth all the way around it. Here's another way to do it, and this is what we'll do in lab. This is the Haemophilus quad plate. So it's a sheep, it's like a one petri dish, but in each quadrant you have a different factor. So in your first quadrant, there's only X factor, or only hemin. In your second quadrant, you only have the V factor, which is NAD. In your third quadrant, you have both factors, NAD and hemin. And then in your fourth quadrant, you have sheep blood, so you can tell whether or not it lyses sheep blood or horse blood. And then it's also got both X and V factor in it. So here we see growth in which quadrants? 3 and 4. So what factors does this organism need in order to grow? Both X and V, right? Is it hemolytic? Can we see hemolytic here? Hemolysis? No. Okay, so which organism is it? Haemophilus influenza, right? How do we know it's not Haemophilus hemolyticus? It needs X and V factor too. The blood's not hemolyzed, right? What about this one? What quadrants do we have growth in? This is 1, this is 2, this is 3, this is 4. 2, 3, and 4. So what factors does this organism require? We've got 2 different answers, X and V and V. Just V. How come? Because it's not growing here, so it's lacking something. This only has X factor. It doesn't need X factor to grow, so it's not going to grow. This here only has V factor, and it's able to grow. So that's what it needs. And it'll grow here in 3 and 4 because they both have V factor, right? Right. So is it hemolytic? Do we see any clearing around the growth here? No. So it's one of those that start with para, right? Because it requires V factor. Is it parahemolyticus or parainfluenza? Parainfluenza? Right. Because it's not hemolyzing, so it's not parahemolyticus. All right. Now, which quadrants would Haemophilus ducreyi grow in? First one, and third, and fourth, right? All right, and again, we do beta-lactamase testing on Haemophilus. So here's our nitrocephin disc, and this is the brand that we'll use in the lab. And you just smear some on the disc, and if it turns pink, it's positive. And what does that mean? What antibiotics can we not use? No penicillin, no ampicillin, no beta-lactam antibiotics at all. Okay, so this is the Haysick group. And we're just going to talk about some characteristics about it. They are fastidious. They're gram-negative bacilli that do not grow on MacConkey. So we know that they're in this group of fastidious gram-negative bacilli because of that characteristic. They're capnophilic, just like Haemophilus. Here they target damaged or prosthetic heart valves. They like plastic. They are a significant cause of endocarditis. So if you have a test question that says, what are Haysick organisms known for? You know it's endocarditis. All right? If you've got a test question that describes a patient that has heart valves, you want to think of these guys. Okay? There are some other organisms that we've talked about that also cause endocarditis. Your coag-negative staph, viridin streptococcus, and enterococcus can also cause endocarditis. So how can we tell the difference between these and your Haysick organisms? What test could we do? Well, quick and easy test. Most common test that we do in microbiology. Gram-St, right? These are gram-negative bacilli, right? And what are these? gram-positive cocci. So that'll help us narrow down our options. All right. And we got our strep. If we got to tell the difference between strep and staph and enterococcus, we could do a catalase, right? Strep or catalase negative, very good. And staph is catalase positive, very good. Haysick. I'll make sure you are able to tell the difference between viridin streptococcus and enterococcus. What tests do you do? Enterococcus is sodium chloride positive and PYR positive, whereas viridin streptococcus is negative. Haysick organisms, these are the, what is that called, an acronym, pseudonym? This is what each of the letters in the group stands for. So H stands for Haemophilus, but of course they had to change the name and make it difficult for us, so now it's Aggregatibacter Aphrophilus. I liked Haemophilus Aphrophilus way better, but what do I know? And then we've got Actinobacillus, Cardiobacterium hominis, Eikenella corrodens, and Kingella. So make sure you know what, be able to recognize each of these. And probably for simplicity's sake, I'll keep calling it Haemophilus Aphrophilus because not everybody's changed over to the names just yet. So Cardiobacterium hominis, let's see, what do I want to know? Big thing here, doesn't grow on McConkey. That's all of these organisms, right? Let's see. This guy shows rosettes on the gram stain. He's why. That's what I want you to know about this guy. So he doesn't color well with the gram stain. He often appears gram positive, especially in this picture. He looks gram positive, but he's actually gram negative. But see how they kind of look like a star? That's the rosette. So I just want you to link rosette and cardiobacterium and Haysick and endocarditis, okay? Normal flora of the nose and the mouth, and most of these are. Eikenella corrodens. Think of this guy, fight and bite wounds. So if someone gets in a fight and you punch somebody in the mouth, you can get Eikenella on your knuckles because it's normal flora in the mouth. And so when you jab them in the mouth, you get an infection. Or cellulitis. Drug users will lick their needles clean. A horrible habit. And they'll get cellulitis where they inject the needle because of that mouth flora that they're injecting. And then it can cause other things. I want you to know of the fight and bite wounds. Think of the knuckles for Eikenella and needles, of course. Licked clean, gram-negative coccobacilli, yellow pigment. Here we go. Two big characteristics for Eikenella corrodens. It pits the auger. Turn that off, and it's kind of hard to see, but it's described as corroding the surface of the auger. So if you want to think of that, corrodens and corrodes, and then it has a chlorine bleach-like odor. So if you got a test question that says chlorine bleach, gram-negative coccobacilli, think of Eikenella. And then your Kingella, no growth on McConkey, catalase and oxidase negative, so that helps us differentiate it from Neisseria and Haemophilus. It can grow on modified Thayer-Martin, so you might have a test question that asks you to differentiate it from Neisseria, gonorrhea. And then Pasteurella. This is an Haysick organism. This is gram-negative coccobacilli. We see bipolar staining, so it won't stain as intensely in the center of the bacilli. It'll stain more intensely on the ends, so it'll kind of look like a safety pin. It does grow on sheep blood agar and chocolate agar, but it doesn't grow on McConkey, so that's a big clue there. It's a zoonotic infection that colonizes the upper respiratory tract and GI tract of animals. So cat scratch fever is this organism here. It's a biochemical test. It's catalase and oxidase positive, and we might see some weak acid production. So think of cat scratch fever when you think of Pasteurella. Brucella. This is also a zoonotic infection. You can acquire it through your contact with urine and milk from animals. So your vets are at risk. People that handle animals, hunters, are at risk. And then your lab employees are at risk. Why are we at risk? Because we're messing with it, aren't we? So we're up there too. It is a potential bioterrorist agent, so you want to work with it under the hood and use extra precautions. It is very contagious, and it's spread through aerosols. These are the different strains. Brucella metelliensis is the most common isolate that we see, but we also see abortus and canis and suis. And basically, brucella will cause abortion. Not only in pregnant women, but also in animals. So you might have a farmer who is dealing with an animal that has aborted, like a cow that's aborted a fetus, and he's touching the placenta, and he can contract the disease. It's called, when you contract the disease, it's called brucellosis or undulant fever. Notice this is bold, right? That's important stuff to know. So what it's described as is a fever that occurs at regular intervals, like every morning they have a fever and then it goes away. And then the next morning they have a fever and then it goes away. And the next morning they have a fever. It's like a wave of a fever. And it happens for days, months, or even years. So undulant fever, brucella. Let's see. Another big, doesn't grow in McConkey auger, so it's a fastidious, catalase oxidase positive. It's also H2S positive and urease positive. I'm not going to make you memorize the biochemical tests here. Mostly I want you to recognize the characteristics of the disease and what it causes. Francisella tularensis. Again, it's a zoonotic infection, gram-negative coccobacilli. It causes tularemia, or rabbit fever. It's another bioterrorist agent. It's very easily contracted and spread. Visible colonies after 72 hours. It's teeny, 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 tiny colonies, and it takes 3 days before you can actually see them on the auger. So that's a big clue that you've got Francisella. It requires cysteine to grow. Notice these are underlined and bolded. Okay? BCYE or modified Thayer-Martin or chocolate auger will grow Francisella, so it won't grow on sheep blood or McConkey. I've got Francis the rabbit pope here. I don't remember why pope. There's some reason why I thought of pope to help remember this. Francis the Pope? Maybe that's why. Francis the rabbit pope. Whatever. I remember it that way. Okay, Legionella. Legionella pneumophilia causes Legionnaire's disease or Pontiac fever. I think that's the less severe form of the disease. And it loves humid environments and water sources like air conditioning systems, respiratory devices that we use in the medical field. So it was first discovered at a Legion, American Legion. Has anybody heard about this guy? So a bunch of old men were drinking and smoking in an American Legion building, and I guess Legionella was in the air conditioning vents, and they all started coming down with Legionnaire's disease, this pneumonia-like disease. And immunocompromised people are at risk, chronic lung disease, of course, at risk for pneumonia, alcoholics and heavy smokers. I put Hugh down because they thought that the Playboy Mansion had a Legionella in the hot tub. Infection ranges from asymptomatic to deadly. It's spread through aerosols. Detection, we can culture it, but it's kind of difficult because it's a fastidious organism. The ideal method is antigen in the urine. So we'll get that all the time. Do you guys do them at Decatur? Do you collect them? You send them out? They'll usually do the antigen test in the urine. There's the DFA, and serology can also be done. So specimen types, of course, it causes pneumonia. So you want to collect respiratory samples like a BAL, where they go in and wash out the alveoli in your lungs with saline and then suck it back out. Bronchial wash-ins, sputum, and then if you're going to do an antigen test, which is your preferred method, it's your urine samples. And here it is. See how tiny they are? You can't hardly even see them unless you know that they're there. They're teeny, teeny, tiny. Very difficult to culture. So your urine test is optimum. Here's a fluorescent test. Fluorescent tests are useful for tiny organisms that are hard to find. Like your TB, they'll do a fluorescent test for TB. If it fluoresces, it's positive. Culturing Legionella, we do have to culture it. We need to use a specialized media. It does require cysteine to grow, so BCYE is your buffered charcoal yeast extract agar that has cysteine in it. Remember, Francisella tularensis also grows on this because it needs cysteine to grow. So this is on your media chart. I think we talk about a couple organisms that require special media. So BCYE, I think of a cheerleader saying BCYE Legionella. That helps me remember and link this media with Legionella. So it'll grow a lot of the usual flora that grows in sputum samples and respiratory samples. And here it is here, this teeny tiny itty bitty little colony here is the Legionella. All right, and that's a little better there. You got to grow it for at least 7 days. So it's very important that if they suspect Legionella and they want you to culture for it, that they let you know, because it's not going to grow on routine media. Bordetella. This is a Bordetella pertussis or peripatussis. This is the organism that causes whooping cough. Has anybody heard of that? Whooping cough? No. I just included this video here. Oh, come on now. It's not going to let me do it. I'm going to have to copy and paste it. Oh, it keeps going without me. Sometimes videos can help us remember organisms. So I tried to include this here so we can think about whooping cough and why it's important that we identify it. Let's see if we can get some. Can you hear it? Hear it? So this is where whooping cough comes from. Very highly contagious. It grows in the ciliated respiratory epithelial cells, so it's important that we get the epithelial cells when you're collecting the sample. You could get false negatives. 90% of exposed people get the disease, but the vaccine has helped to drop this number, but it's very contagious. And then most of the time it's from adults who are carrying the infection and spreading it to people that haven't had the vaccine, like Pedro. Very serious infection in young kids, and it causes a repetitious coughing followed by a rapid gasp for air, and that creates the whoop. They can have cyanosis, so they can actually turn blue because they're not getting air. And then sometimes they need assistance opening the airway, so a tracheotomy. Probably 10, 15 minutes. The convalescent phase can begin at about 4 weeks after symptoms. So this is when they're starting to, the IgG antibodies are kicking in, and the symptoms may subside, but the coughing can last for months without resolution. Diagnosis and isolation, we want to get samples from the nasopharynx. So this is a good test question. What specimen do you collect for pertussis? You want to get as deeply as possible in the nasal passages. You can also get aspirates. You want to inoculate a transport medium. Reagan-low transport medium is specifically used for Bordetella pertussis. So this is on your media sheet. You want to link this transport media to pertussis. And then you have to inoculate Bordé Jingao. I love saying that. It's a potato auger with glycerol and sheep blood. And this is what we use to grow the Bordetella pertussis. So link this media with pertussis. And then you can also do your routine augers for your routine respiratory pathogens. You want to incubate it at 35 for up to 7 days because it can take a little long to present itself. And you can see it's teeny tiny colonies. They look like mercury droplets. It's kind of a crummy picture. But you can do serology or a DFA antibody test to confirm diagnosis and use that instead of culture. And ideally, if they do suspect pertussis, they should use serology instead of a culture because laboratorians are at risk. And it's much more sensitive method rather than the culture. Okay? So we talked about some specific characteristics for gonorrhea and Moraxella and Haemophilus. And then we talked about quite a few other organisms, but I just picked out some notorious characteristics, some important characteristics. I don't necessarily want you to know what they look like on media, what biochemical tests to use to identify them, but I tried to highlight and underline what I want you to know from those organisms, okay? All right, they're not very common in the micro lab, but they're good test questions. Any questions? When you are memorizing these organisms, a lot of people have said that it's easier to memorize what's positive rather than trying to memorize what's positive and negative. So if you can recognize what's just positive for that certain organism, it kind of helps you narrow down what you need to look at. Okay? You can always email me if you've got any questions. Next week, we're going to do some learning activities to help reinforce the last 4 chapters we've talked about to get ready for the exam. So exam 3 is chapter 17 and 18 and 14 and 15, right? So this is a big test. Yes. 14 through 18. So make sure you start memorizing those organisms and how to identify them, what they look like, characteristics. Use the questions in the back of the chapters to help you. Use those resources. A lot of them have quizzes. Quiz each other. Use each other. Study groups. Okay? I did, under next week's content, I put in some assignments that you can take as many times as you want to just kind of quiz yourself and see how well you're doing with your memorization of the material. Here we go. So you can't see them quite yet, but it'll pop up on Wednesday. And if you want it sooner than that, I can open it up for you, but I didn't anticipate anybody ready to take ready to undergo these assignments. So I got gram-positive cocci and your fastidious organisms. Quiz there. There's a case study if you want to take a look at the case study and answer questions. Print them with you. We can work on them in lab. If you want to work at them at home, that's fine. But just make sure you come prepared so that you're not starting from scratch next week. Make sure you get started on your study and don't wait till the last minute. Okay? And then we're going to play some bingo. I've got a review game so we can quiz, see how well you're doing with your memorization and identifying organisms. And then if you've got particular questions, you want to go over a certain concept, bring that with you and we'll go over that. Okay? All right. That's it.